My name's Mark Silverberg. I am the PI of the University of Toronto GRC, part of the NIDDK IBD Genetics Consortium, and our consortium is happy to host periodic webinars on topics of interest to our group, as well as the broader scientific community that's interested in the study of inflammatory bowel disease. It's a pleasure to welcome our speaker for this afternoon, who's a colleague here in Toronto at the Lanthanide Tannenbaum Research Institute at Mount Sinai Hospital in Toronto. His name's Hartland Jackson. Hart is Canadian. He did his PhD at the University of Toronto and subsequently did a postdoc at the University of Zurich in Switzerland in quantitative digital pathology and systems biology. Hart's new to the Lanthanide about 2 years ago, came on staff here, and we're very excited to have him engaged in contributing to the work that we're going to be doing on inflammatory bowel disease. His expertise is in imaging mass cytometry, where he's done some of the seminal work on both the computational side as well as the technical aspects of doing high throughput systematic approaches to large scale tissue analysis, initially in the breast cancer field, but and now hopefully focusing a little bit as well on immune mediated disease and intestinal tissues. And Hart's also already at this stage of his career received quite a few awards, including a Canada Research Chair in Systems Pathology, an Ontario Institute of Cancer Research Investigator Level 1 Award, and most recently the Terry Fox Research Institute New Investigator Award. So Hart, thanks for joining us, and I'll turn it over to you.
Thanks, Mark, and thanks for the very nice introduction. I think you let the cat out of the bag at the end of the presentation, which is I've tricked you all into joining a cancer presentation, and then we're gonna change directions and talk about, hopefully have some good discussion around future applications for inflammatory diseases using this technology. And so, as Mark mentioned, my focus has really been on kind of looking at tissues as a multicellular organ, tissues or tumors, and not just as individual biomarkers. And for this, my past work in my group are kind of focused on the use of imaging mass cytometry and combining it with other methods. So I'm going to give you kind of a good background in imaging mass cytometry itself, how we've used it so far in breast cancer, some of the new work coming from my new group here at the Lanthanide, and some quick images to show you why we think this has great potential in inflammatory bowel disease. So So as I mentioned, my group is mostly focused on the study of cancer. And like many diseases and those that you're all studying as well, these issues are arising within healthy tissue. And we need ways to distinguish the diseased tissue from surrounding healthy tissue. And a lot of the time, this is done by pathologists looking at the organization of the tissue, or looking at alterations in the types of cells that are present there, such as immune cell influx. But there's no individual marker that's really going to address the complexity of anything that we're looking at. And so we do really need to be looking at multiple parameters all at the same time for multiplexed marker expressions. And with the capability to look at multiple markers, we are looking at many cell types at the same time, many cell subsets in the different immune hierarchies. And also the functional states of those cells. For example, we do a lot of tumors looking at what signaling mechanism each type of cell is undergoing, or the exact cell state. But beyond this, when we can see all the cells in one location or one image, we can actually start to look at how groups of cells are coordinated, what neighborhoods of cells are present in these tissues. And we're interested in using these as potential biomarkers, or to understand kind of cellular networks that are working together to lead to specific tissue functions and what goes wrong during disease development. So to get at this kind of multicellular function and neighborhoods features, we know we need a technology that has single cell resolution, needs to be spatially resolved, we need to be able to measure lots of things at the same time. It needs to be quantitative for all these features, but also importantly needs to be compatible with clinical procedures and clinical tissue samples. And so for this purpose, imaging mass cytometry combines the spatial resolution of a laser ablation system with the multiplexed measurements of a CyTOF ICP mass spec. And so CyTOF technology was originally brought out as the competing technology for, let's say, for flow cytometry. And now with imaging mass cytometry, we're seeing this as kind of comparative to immunofluorescence or microscopy. The difference being is that, you know, these standard flow or microscopy are based on fluorescence-based tags. So antibody-based measurements, but with fluorophores attached to each antibody. Whereas in CyTOF or mass cytometry, we're using the same antibodies. The difference is each of our antibodies is labeled with a rare earth metal, a metal of very specific isotope. And we're then measuring those isotopes on a mass spec. The benefit of this is that unlike fluorophores, which have kind of very broad emission spectrum that can run into each other and cause the need for different computational methods to deconvolve the different signals, the CyTOF, you can get access to quite pure isotopes, and the mass spec itself has very good resolution to discern different metals in this case. And so we're able to use many, many more channels than what happens when you overlay the emission spectra on fluorescence. Beyond that, with these different fluorophores that you're using, there's also very different strengths of different signals, hard to compare across different channels, whereas we can actually really control the exact same number of metals and look at how many of those are actually physically hitting the mass spec and getting counts there. So while I'm saying that we're looking at metals, mostly this is the Lanthanide series of metals. These are rare earth metals, but in actual reality, they're not actually that rare. They're pretty abundant. They're quite safe to work with, which is nice. They're non-radioactive. And a benefit is they have very similar chemistry. So we can link these to the same type of polymer that we can conjugate to our antibodies of interest. Another benefit is these kind of high mass elements are not ever present in any biological samples. So there's very little background. And this leads to our ability to quantify signals quite well. And while at the moment we're in about the 40 to 50 marker range for imaging and over 50 for suspension CyTOF, the TOF itself is actually able to measure probably 200 different mass range of 200 different AMU. So to be able to look at more isotopes as they become available or as the chemistry improves to link these metals to antibodies. And as I mentioned, there's very little compensation needed between the different metals on these samples. And another big benefit is that, you know, issues such as autofluorescence that are going to happen in many fluorescent-based methods are not present here, as there's no light signal. You're not going to get kind of background fluorescence from any of your tissues. So the staining procedures for imaging mass cytometry are very, very similar to standard immunofluorescence or immunohistochemistry protocols. The difference is that the antibodies we're using each have these metal tags on them instead of a fluorophore or a HRP, horseradish peroxidase. So once you've completed kind of your staining protocol, you will stain with one cocktail of all 40 of your antibodies all at the same time. And then after this, you actually dry out your tissue section, which is a little terrifying to anyone who's used to fluorescence methods, but is what's actually needed here. And then we introduce the dried sample that contains all the antibodies stained into a laser ablation chamber. So in the chamber, the laser then actually rasterizes the tissue. So the laser pulses across the tissue. It ablates a 1 micron spot of tissue, and this is aerosolized and pushed in directly into the plasma of the mass spec, where it's completely atomized down to its pure elements. These are then filtered in the mass spec to look at just the high mass elements that we're interested in. And then these are read out on the time of flight mass spec. So for every laser shot, we're then doing one measurement on the mass spec. And then we use that one measurement to make that an individual pixel in an image that we restructure. So the one laser shot destroys the piece of tissue. It leads to one measurement, and that measurement becomes a pixel. And as we reconstruct an image. So over my time using imaging mass cytometry, I landed in Zurich at the Bodenmiller lab, and they had just kind of had the prototype of the first imaging mass spec system. And so what this was a laser ablation system from the geology department had been rolled directly in front of a standard CyTOF, which was used for suspension measurements. And then if you can see my mouse, there was this very just direct line rolling through to introduce the sample off of the laser ablation system straight into the TOF. And so originally we were able to do 32 markers, and the laser speed was about 20 hertz, which meant for a tissue microarray, or let's say a punch biopsy would be something that you'd be regularly using. It would take about 3 hours. I thought I was, great at pretending to be an engineer, and I had optimized gas flows to get us up to about 40 hertz. But right about the same time, the whole system was actually commercialized by the company that produces cytofs called Fluidigm. I think actually they've just changed their name to Standard Biotools, but they now produce this imaging mass cytometry as a commercial product. It's able to do 50 markers. Current standard is about 200 hertz for about 20 minutes. And this recently, with new modifications, this is now going to be doubled to about 400 hertz, meaning that each image we're looking at about 10 minutes for a teammate core, a 1 millimeter squared region, let's say, and that this is going to be all 40 plus of your antibodies all simultaneously at the same time. And so what this actually looks like, you can see here, this is an actual image and video off of the cytof as it's running. So along the left, what you're seeing is actually the laser running across the tissue section, and the white square box or square that you see is where the laser's already ablated and removed all the tissue right down to the glass. And then if you look carefully, you can see it's just carving off the next line in the image, that next row, which is going to be this one, so it's pulsing 1 micron spots all the way across, just carving off that piece of tissue. And then this is being digitally reconstructed into the image on the right, where we're measuring out all the different markers that we're interested in looking at. So one of the first experiments that I was doing in Zurich was to actually directly compare imaging mass cytometry to immunofluorescence. And so I did this by staining both metal tagged antibodies and fluorescent tagged antibodies on the same sample, doing microscopy, and then taking the same sample and putting it into the imaging cytof system. So the staining strategy you can see is here on the right, where the primary antibody had one specific metal tag, and then there's a mixture of 2 secondary antibodies, one that was fluorescent and one that has a metal tag. And we did this kind of at the limitation of standard microscopy, so 3 different fluorescent channels. And so for each primary stain, we're actually getting 3 measurements. We're getting 2 cytof measurements and 1 fluorophore measurement. And that's what you see in these 3 colors. So looking at HER2, which is a receptor tyrosine kinase used for breast cancer diagnosis, or pan-cytokeratin to identify all immune cells, or our DNA interchelator, metal or DAPI, we're able to see that all 3 of these measurements for all these things at the same time. And so if you look top to bottom, you can see that the overlay of fluorescence in microscopy is almost identical. That's why it's end up being white for either any HER2 pan-cytokeratin or the DNA interchelator. But what you can also see is that this image here on the left, this one actually has some red going on, is because there is a lot of collagen and matrix proteins here that are leading to autofluorescence. And it's showing up red because we only see this with the microscopy, but we don't see any of the autofluorescence present in the cytof-based measurements. So alternatively, you could use fluorescence or either of the cytof measurements to basically reconstruct very similar images of this exact same piece of tissue. And if we zoom in now on this specific region here, you can see directly kind of what the resolution is of IMC versus immunofluorescence. So a standard microscope, we're probably looking at about 300 nanometer resolution, whereas the imaging mass cytometry is a 1 micron resolution. So you can see it's a little blurrier than the microscope, but you can also see that this is more than good enough to do subcellular resolution to identify single cells. And while the image on the left kind of maxes out at about 3 or 4 channels, our standard IMC panel at the moment is 45 or more markers. So the multiplex capacity is much, much higher. So as I'd mentioned, the quantitative capacity of the machine is actually quite good. This is because we know we're measuring that exact depth of the tissue that you've cut, and we're getting all of that material in there, and we know it's a 1 micron spot. No autofluorescence, really reproducible. Part of this is because we can simultaneously stain all of our antibodies at the same time. There doesn't need to be cycles of strip and stain as what's necessary for other multiplexed methods. And an interesting feature is that these stains are actually quite stable because we don't have a fluorophore in here or an enzyme that can degrade over time. So you can do your stain and then kind of leave it at room temperature. In a case for, we've done it for over 6 months, up to a year, and the stains are still great. You can still go back and image them at any time. So they're quite stable. Challenges of using this system are it requires having a cytoff of yourself, which is basically the cost of a mass spec. It can be more expensive. Well, we're kind of in the 40, 50, 60 marker range. The limitations of this are what pure isotopes we can get access to. And so some of that is just, these super rare other metals would provide us other channels, but costs or chemistry to bind them to the antibodies is not available as of yet. And because we're actually, 1 micron by 1 micron rastering this tissue, it can be maybe slower than a really fast microscope that would be able to tile across a whole slide really quickly. And then another kind of challenge is these are all antibody-based measurements. So you need to have really well-validated antibodies, and you need them to all be working in the same staining protocol at the same time. There's no optimizing this antigen retrieval method for one antibody and another, because what sometimes happens is you improve it for one marker and you're going to lose others. So you kind of want everything working under the same protocol. So some of the, application benefits of this is that there's no tissue dissociation. We're getting single cell data from tissue sections. These have come from biobanks. These are formalin-fixed paraffin embedded samples that every hospital is already producing. And there's also less processing, and there's almost no sample loss. You take your slide and you put it in the system, and you're going to measure every cell that you hit with the laser. So this allows us to do really small samples and get all the single cell data from it. So for example, punch biopsies for IBD, things like circulating tumor cells or other biopsy samples where dissociating those, you're going to get a lot of tissue loss and not be able to do some other, or not have enough material for other measurements. They're quite nice for this. That same feature can be applied to kind of larger samples where we are doing, upwards of millions of cells in a data set rather than the tens of thousands. Just depends on how many you're gonna image. So to get you an idea of what these images look like, this is an IMC image of a breast cancer. So here you're looking at the counterstain reagent that we use, ruthenium tetroxide, which is going to bind metals to any protein in the tissue sample. And then what you see is on the right, these black circles are the fat lobules of the breast, and the tumor mass is on the left and kind of invading and pushing into the healthy stroma. So I'm going to show you a couple markers. These are in combinations of 3. The reason being is that one, we're used to looking at 3-color IF images. The other is we're not able to see 40 colors. It kind of really quickly past 6 or 7 markers loses all meaning. But so every group of images that I show you here is all from the same sample. It's all from one stain, one tissue section. And so what you can see in here is that, we can use markers that are identify the function of cells. We can see which cells are mitotic. We don't have to just measure the epithelium or immune cells. We can also look at matrix proteins and non-cellular features such as fibronectin here, but also look at the architecture of different and location of different cell types, such as myoepithelial cells surrounding or encasing epithelium in this case. It is possible to do epigenetic marks and post-translational modifications, such as H3K27 trimethyl mark, which is here in blue. And then a really nice feature that I think we're interested for IBD and is kind of one of the most popular uses of the technology is to look at a different immune cell types in different locations. And what's nice, I think, is that we can see these immune environments, but you could also look at what they're interacting with. So in most of our studies, we're looking at the immune phenotypes, but also what type of tumor they're associated with and what's happening in those cells. And this is really revealing way more heterogeneity in all the tumors than we're looking at than we would like, but that's the complexity of what we're dealing with. So to analyze these samples, what we do actually is we take all the high dimension images and we segment these images using machine learning methods that identify the boundaries and locations of all the cells in the image. So from this mask of the different cell locations, we then quantify all 40 markers from our IMC stain, but also the visual image spatial features of these cells. And we put this all into a single cell data set. So these single cell data sets will include your 40 measurements, but also things like the area and the size and the shape of the cell, or how much of its membrane is touching other cells, and even the identity of these other cells that it's interacting with. And then we can use all of this information to start to define different cell types that we've extracted from these images and look how they're organized. So we'll do this using similar tools to what's being done with a lot of other methods, including single-cell RNA-seq or other high-dimension methods. So in this example, we're looking at a t-SNE map of the single cells coming from 310 breast cancer samples. So each dot in the t-SNE plot is corresponding to an individual cell that was in one of these images. And then they're localized on the dimension reduction plot so that individual cells that are more similar across all their parameters are physically closer to each other in these plots. We then clustered these according to the marker expression to identify different types of cells. And that's what you're looking at with these colors. So all the rainbow of colors is mostly different types of tumor cells, but we also see multiple types of fibroblasts in blue and different immune cell phenotypes in green and endothelial cells in yellow. So this measurement took place for over 310 biobank breast cancer samples. And what was nice is that some of these samples were over 20 years old. And so we actually knew long-term follow-up what had happened to these patients and could retrospectively investigate the cellular contents of these tumors and how this related to outcome. So each stacked bar that you're looking at here is the quantification of different cells from one of these breast cancer tumors, where again, all the different colors are tumors, blue are fibroblast populations, green immune cells. And if we had looked at how these are classified in the clinic for breast cancer, they're kind of classified into 4 different types. But if we classified them by the cell content, we were looking at over 18 different types of breast cancer. And so for all these cell types, we're not just counting them though, we actually know where they are in the images. And what we could see is that when we place these back on the images, there's also, even for the same type of tumor cell, many different types of organizations were taking place. And so to really quantify this organization, what we did is we looked at the location of every cell and the identity of its neighbors. And this kind of built a network of which cells are physically interacting with each other in the tissue. And we analyzed this using modularity scoring methods that would be similar to what's used to kind of identify groups of friends on Facebook, for example. And so, but in this case, what we're using it for is to identify groups of tumor cells that are interacting and touching each other, or distinct environments. So both the tumor patch, but also the surrounding immune and fibroblast cells that were interacting with each of these. And so what was interesting is if we did our single cell phenotypes associated with outcome, these were not related to outcome in patients, but the multicellular features that we saw actually were related to outcome. And even in some cases, it was the organization of these cells, even the same cell type, that would be more linked to good prognosis or bad prognosis. And this would even flip for some samples. So some tumor types, it was better if it was a small patch, and some tumor types better in a large one. We also then could see different tumor microenvironments, such as blood vessels containing distinct types of immune cells could be related to bad outcome, for example. So this also allowed us to do kind of spatially resolved measurements. As if we do the imaging in different areas of a tumor, we could look at the spatial heterogeneity and cellular content in different locations. And so on the left, this plot that you're looking at is 72 tumors where we had 4 different regions from each of the breast cancers. And then each dot was classified by the cell contents of what it would be. And we scored the spatial heterogeneity from spot to spot using Kublik-Leibler divergence, which compares each location to kind of the average of all locations. And so a larger measurement means it's more distant from the tumor average. And so a lot of them, the spatial heterogeneity was very low, but there was a small subgroup of tumors where there is a lot of variation. And we could identify that this was a specific subclass of breast cancers that actually had more spatial heterogeneity. And so this would be kind of interesting if you are doing a diagnosis based on a biopsy. You kind of want to know how representative is one biopsy of the multiple locations in a tumor. So going further, in a second paper, we associated this with specific genomic aberrations. So we have all these cellular contents, what the cellular phenotypes of these tumors are. And in this metabrick cohort of patients, this has been steadily developed as a nice resource in the breast cancer field, where they have sequencing of oncogenes and copy number aberrations, which were driving different subgroups. So getting access to tissue microarrays from this same cohort, we were then able to use imaging mass cytometry to look at what cell phenotypes are present in tumors and link those to the genomic aberrations in those same tumors. So in this plot that you're looking at, each row is a specific genomic aberration, such as a mutation or an amplification or deletion. Sorry, I got that wrong. Each column is the mutation, and each row is a single cell phenotype we identified by imaging mass cytometry. And then the pink spots are positive associations and the green spots are negative associations. And if it has a black circle around it, that indicated that by linear regression, we could see that these were highly associated with each other. And the black dots are indicating statistical significance, whereas kind of the outline of the spot was how often we saw that in this data set of about 400 patients. And we did see things that you would expect, such as, for example, hormone receptor positive tumors were associated with PI3 kinase mutations, and P53 mutations were associated with the more dangerous triple negative breast cancer phenotypes. But this also revealed some interesting things that we wouldn't have expected, such as hypoxic cells seem to be linked to immunomodulating genetic aberrations, including PD-L1 amplification, which is here, and beta-2 microglobulin deletion. Which is here. So this is something that would be pretty interesting to follow up on, as I haven't found a lot on this relationship between hypoxia and immune suppression, for example. So together, this is kind of had set up kind of what the purpose of my postdoc was to basically use the multiplexed imaging technologies to look at the kind of the sum of all the cellular components in a tissue and use that for kind of what we are calling single cell pathology. How, if we analyze the single cell features of a tumor, does that provide us more information for survival? And in breast cancer, it was able to identify new subtypes of breast cancer with distinct outcomes, and also spatial features, and show that spatial features in the organization of the cells are specifically related to outcome. So, in a topic that maybe many of you are more interested in, how we're continuing this is not just to have breast cancer specific panels focus, but to make whole 40 antibody panels with specific questions in mind. So for example, Eddie Chen, who's a postdoc in my group, has now developed immune profiling panels where we're able to look at multiple T cell subsets, multiple myeloid compartments, all at the same time in one image. And so in this case, we're looking at a mouse lymph node tissue. And within the individual lymph node, we're then able to look at T cell subpopulations on the left, or other cell subtypes or myeloid compartments on the right. And so combined, what he's done is kind of made these paired matched human and mouse immune profiling panels. One panel that we can use in human tissue to look at immune cell subsets, and another for mouse tissues. And our goal is really to be able to have multiple studies where we have both mouse models and human tissues, and we're able to actually model these systems that we're looking at, and not just do associations with clinical outcomes, but to functionally challenge these in tumor models in the future. So one example of this was to look at pancreatic cancer, which is a subgroup of my group is interested in. We're able to look at different subtypes of pancreatic cancer. These have been defined based on their transcriptome as either classic, or the more dangerous basal subtype, or mixed and in between. And if we then apply this immune cell profiling panel to these, and then segment and analyze the tissues using tools such as TISNI and clustering to look at different marker expression, we're able to look at upwards of 30 different immune cell types, specifically in these tumors, and how these are spatially organized in these tumors. And what we've seen is, based on the transcriptional profiles of different subtypes of tumors, there's actually different tumor immune microenvironments present. In these classics ones, this is much simpler, really being more focused on myeloid cells being present, or NK cells. Whereas the basal cells, which everyone says, oh, those should be a little more inflamed. Yes, there's more cells there. There are T cells present, but these are also very interacting with different myeloid-derived suppressor cells or other phenotypes. And then there's a lot more heterogeneity taking place in the mixed tumors, more immune cell types present. And so we're continuing this to look at kind of what can you do when you immune profile all these different cell subtypes in the tumor environments. And what we're looking for in one case is we're looking for someone to come in to kind of not just look at counting immune cells, but to look at what environments these immune cells are present in. So in serial sections or in the same tissue, we can not just say where is that immune cell, but what is that environment? What's the metabolic state? What's the matrix surrounding this? What type of epithelial tissue is it interacting in? And to look at the immune environment in a specific location. So we have a couple different collaborations to investigate this kind of immune profiling studies, many in response to immunotherapy. A specific example here at Mount Sinai Hospital is to look at sarcoma response. And this is a really nice image that we have from an undifferentiated pleosarcoma, looking again at immune profiling panel. You can see in this tumor, all the green are all the immune cells and that they've kind of been excluded from the tumor center. And then a lot more vasculature around the outside, and also really nice important spatial features like this tertiary lymph node structure, which we see here. These have more recently been shown to be linked to response to immunotherapy. And what we can now see is there's different types of tertiary lymph node structures. But at the same time, in these tumors, there's a lot of diverse suppressive cells. And so in our mind, it's not really enough to say, yes, there's T cells present in these tumors, so they should get it. It really depends on the context of where these are and what types of cells they're interacting. So Eddie is going farther than just wanting to count all the cells and identify their clinical associations. And he's wanting to look at how functionally do epithelial tissues influence their environment. And so one example of how he's doing this is in the mouse mammary gland. We're activating a specific pathway in the ductal epithelial cells themselves. So the cells with green, we've turned on the hipposignaling pathway, and you can see that it's present in those ducts. And either on controls on the left or in the pathway activated on the right, we can then look at what that does to the surrounding environment. So if you alter the epithelium, how does the immune system respond? What's happening in the tumor immune microenvironment surrounding that ductal structure? And so we have a couple interesting models to do this for cancer, but I think this would be pretty interesting to do in inflammatory diseases as well. Modify the epithelium, but use spatial measurements to look at what's happening or what happens surrounding it as a response. So overall, the lab is really kind of interested in these multicellular structures. How are cell signaling networks taking place in healthy tissues, and what goes wrong in the cellular communication as this progresses towards disease? We do have a focus on cancer, but also think this is pretty important in inflammatory disease. And so we have done some initial pretty exciting just pilot studies with the IBD group here in Toronto, and we're interested to do more of this. We can see, pretty extensive immune infiltration in this punch biopsy of an IBD patient, and that it does seem to be pretty nicely localized in these certain phenotypes in specific points. So we are looking for someone to come and join the group that would be excited to work on IBD and inflammatory disease with us. You'd be joining this very hyper collaborative group that we've put together. We're only just barely 2 years old. We are 10 people, and we're still looking to grow. We need a couple more along the way. But there's lots of tools now up and running, really exciting time in the lab. I think it'd be great to join. I also think this IBD project where we're looking for someone is pretty key. As a PI, I have all the resources to provide and technology expertise on this point, but this isn't necessarily, this is something that I would want someone to be pretty independent on, to be collaborating with the clinical teams here in the hospital and to kind of forging their own path. So I think it would be a nice, very independent project with a nice future direction for a new group to do spatial measurements in IBD. So if you're interested, please get in contact with me. I'm also excited to discuss what we're doing and answer any questions that anyone here has as well. Thanks for inviting me.
Great. Thanks, Hart. That was an amazing overview. And I think you could see the real exciting potential of making use of the massive amount of histopathology that's in a lot of our centers and how it can be incorporated in our work in IBD.
I think the ability to go to these biobank samples is
huge.
Very exciting.
So I'll start with reading out some of the questions in the chat, and then happy to, if anybody wants to ask some live questions. But so there's a question: do you think there will be a possibility to perform IMC and MALDI MSI on the same tissues?
Yes. So what has been done is DESI mass spec imaging can be done on the same section, but there's a lot of groups, us included, doing mass spec imaging on serial sections with IMC. So while you're going to get thousands of peptides and very different information, the IMC does provide this ability to amplify certain signals and to get single cell resolution. So one of the things that we're doing is mass spec imaging on one section, and in a serial section identifying the immune cell populations that are present there. And then because we can perfectly overlay these, we can actually link the immune cell types to specific metabolic environments or to specific environments as identified by the mass spec. So I think that's really exciting. I think multi-omics in terms of combining this with other technologies is something we're very excited about, and each one's providing its own important data set.
Great. Can you quantify subcellular signal intensity for membrane, cytoplasm, and nucleus?
So it's a little difficult because what you're actually looking at is a compressed Z stack of your whole tissue section. And so you can get small overlaps between cytoplasm and nuclei. And so maybe on a per pixel level, this isn't perfect. We are working with some collaborators here at the Linfeld, Kieran Campbell's group, and they have some pretty fancy methods, so that there is some hope to be able to distinguish these things more accurately. I would say we can do nuclei to membrane quite well. To say cytoplasm versus membrane, it was probably a challenge as these things are overlapping. And it also really depends on what type of cell you're looking at. If we're going to talk about, a nice big fat tumor cell with a clean border, then this is probably much easier than a very small T cell with lower resolution, or with our resolution, that's a little bit tougher, but it's possible for sure.
Okay, this is a question that you and I have talked about a lot, is how do you how old samples can you go back to, and how does it matter how they were stored and how they're prepared?
Yeah, so yes, yes, and yes. What I would say would be the thing. It does matter how they're stored, and that's a big challenge that you don't always know when you get them in your hands. We prefer to get fresh cuts is ideal, but a good thing is that the proteins seem to be much more stable than RNA-based measurements. And so there is the benefit that antigens are still working quite nicely in many cases. One of the things that we're working on at the moment is we're building in kind of internal controls to look at total tissue stain, and you can compare, because you're looking at multiple markers, kind of relative expression of different markers in these cells, and get an idea of if there was an issue with a specific stain or marker. And that's because you're doing 40 at the same time, and rather than just individual one-offs, if it worked or not, you're not so sure. And that builds into how you quantify these things as well. With multiple measurements, then you can look at relative signal expression rather than just absolute counts in order to identify and quantify what you're looking for. So I'd say it is possible to do extremely old samples. It does depend on how they're stained, but from what we've seen, we've done 20-year-old FFPE blocks that have been quite successful for staining, and this would be the same issues that you'd run into for immunohistochemistry or immunofluorescence as what we're doing here.
Great compliment, great talk. Can this detection methodology be applied to in situ hybridization, and has it been applied to look at spatial organization of microbial ecosystems?
That's a super cool question. So I haven't seen the microbial application yet. It does work with in situ hybridization. So there's a cell systems paper from Daniel Schultz is the first author, where they adapted ACD RNA-scope system to put a metal tag on it instead of a fluorophore. And then they're able to do the in situ hybridization for RNA with IMC. The exciting part of that is that he was able to do, now at this point, ACD's extended their number of channels that they can do. And so you're able to do 15 to 18, I think, in situ hybridization probes. But then if you still have an extra 20 plus markers, channels available on the CyTOF, you can fill those in with antibodies as well. So you can do in situ hybridization and antibody stains on the same tissue section, which is pretty nice. And so it is possible, and I think that those kind of methods are going to get better and better as more people are working on them too.
Great. Have you attempted to design panels of ligand receptor pairs to define functional microenvironments?
We would love to. So, I feel like I'm just pumping Daniel Schultz, which is he deserves it. So it should happen. But so Daniel's also got another paper. Him and Tobias Hook have a recent paper just out that what they've done is a serial panel to do in situ hybridization to get the ligand. And then some cases are looking for receptors. The major challenge is to do immunofluorescence staining on ligands can be difficult because they get secreted and then they are diffused. And this is why, for a lot of different systems, either in flow style methods, you're blocking secretion so that builds up and then you can measure it by an antibody. So it's really ligand specific of whether it's going to work in an imaging based stain. And some do really nicely and some are just a major challenge, but in situ hybridization can be a way around this. So in terms of like whole network profiling, I think that'd be a nice way to go to kind of take single cell RNA-seq and predicted receptor ligand pairs, but to see them spatially. I think that's a really nice question. The earlier question about the microbiome as well to come back to, I think in situ hybridization to identify what's there would be pretty exciting, especially because then you could look at what's the surrounding immune response or epithelial integrity at that same location. So that'd be quite nice too.
And just a comment from Suzanne Devkota, Cedars-Sinai, we have successfully performed 16S FISH and IMC on the same tissue. Happy to share the protocol.
I would love it. That's
okay.
Great.
I can put you in touch.
Perfect.
Any other questions live that anybody has? Feel free to either put up your hand or just shout out.
Hi, Hart. I wonder if I can ask a question. It's a bit of a technical question, but although it's hard to say, there's such limitation when you have 40 possible markers, it's probably you could do many things. But as you say, some antigens don't work under certain conditions, and even some combinations of markers, you sort of run out of space. So I'm just wondering if the issue of looking at slices, contiguous slices, and can you compare or analyze between slices?
Yeah, so it's you're let's say normal section, you're about 5 microns away. So in many cases, you are getting half of the other half of the cell. And so you can align cells, but you're not getting all of them. I'm aware of other groups working on ways to kind of align this quite nicely. What we've kind of been doing is to look at larger features. So while, you might not necessarily get the same cell to let's say to know you're in the same crypt or you're in the same spatial feature is quite easy. And so you can kind of do serial section measurements between different technologies or between different IMC panels and still align the data spatially on a, more macro level, more tissue architecture level rather than single cells. Where, I think that comes into how you design your 40 marker panel as well, is that you want in one panel, you want the markers that you have to see in the same cell. Whereas if you're okay with it being 5 microns away, you might as well, and you have the potential to do it, then you could just do a serial section and another stain. I think a lot coming too, there's people doing the multiplex methods with spatial transcriptomics and other things. And I think that'll be interesting to have your target markers and maybe use those to link across different omics methods, or to go from, broader measurements to single cells. I think there'll be interesting methods coming in the future for that too. But I agree. I think, everyone's used to imaging and, oh my gosh, you can do more than 4. And then within like a couple months, it's like only 40, come on, like we can do more. And you very quickly creep up, and you want more and more and more. So that's not necessarily a bad thing, but yeah, it's not a whole transcriptome method or things like that. You have to ask a specific question and know what you're asking.
So follow-up, does the signal integrate across the entire thickness of the section? Yeah,
so the laser ablation ablates and aerosolizes the whole Z stack. There's no Z information at all. It's whatever section is on the cut, we laser ablate and is gone. So while we're 1 micron in the, let's say in the X and the Y direction, we're maybe 4 or 5 microns in the Z plane. And that's all just mashed together into one measurement. There's no Z. That being said, there is a 3D IMC methods that have now been published by the Bodenmiller group in Nature Cancer. And so doing serial sections, you can start to put together 3D data that way, but that's very image and computational and resource intensive to be doing that.
All right. Any other questions? Okay, well, if not, we'll give people a few minutes more of their day. Thanks a lot, Hart, for doing this. It was great, and we'll look forward to having you back to the whole group, maybe in a year or two, to share the progress that we're making in this area. So thanks very much, and thanks everyone for attending.
Thank you.